So hello everyone, our today's topic is on physiology. So let us discuss the first chapter question. Which of following it is involved in cell adhesion and it is composed of transmembrane glycoprotein? The answer would be cadherins. Well, the cadherins, remember these cadherins, they are number one, transmembrane glycoproteins. So what are cadherins? So these are glycoproteins that play a crucial role in the cell adhesion. So they play a role in cell adhesion. So they are involved in formation of adherence junction, which are cell-to-cell junction that provide mechanical strength and facilitate the communication between the cells. Cadherins, they are involved in cell adhesion and they are composed of which protein? Transmembrane glycoprotein. So imagine you have a bunch of building blocks. Cadherin, they are little connectors that hold the blocks together. So they make sure the blocks, they stay attached to each other in a right way. So similarly, you have a stack of building blocks. So these cadherins will make sure that these blocks stay attached to each other in a right way. Similarly, in your skin, these cadherins would help skin cells to stick tightly to each other. So if your skin does not, so that your skin does not fall apart. So cadherins, they are basically the cellular glue. What are cadherins? They are the cellular glue that ensure that our body cells, they stick together and work as a team. So cadherins, they are like glue that helps the cells to stick together. They are the proteins that are found on the surface of cells. So they use, remember, they use calcium ions to work properly. So when the cells, they have same type of cadherins, they stick together more. For example, one cell that has, suppose one cell has a cadherin, and it will stick together to other cells that will have a cadherin. So think of cells as people and cadherins as the handshake that makes them friends. So if two people use the same handshake, they will become close friends and hence they will stick together. So if the people use different handshake, means different cadherins, they still can be friends, but they might not stick together as strongly. So these cadherins are just like the handshake that helps the cell becomes friends and stick together. Whereas the other option, microfilament, they are composed of the protein. The microfilaments, they are composed of protein that is actin. So they play a role in cell structure, cell movement and cell division. So the microfilaments, they are involved in process like muscle contraction, cell motility and maintenance of the cell shape. Microtubules, they are other component of cytoskeleton. They are made up of tubulin protein. So these microtubules serve as track for the motor proteins like kinesin and dynein that transport the cellular material. Whereas the intermediate filament, they are composed of various type of proteins like keratins, pimentins, etc. So they will provide mechanical support and maintain the structural integrity of cells and tissues. So which of following cells they are involved in cell adhesion and it is composed of transmembrane glycoprotein? The answer would be cadherins. Hope you understand that. Let us move to the next question. The next question is which of following organelles, cell organelles, it is responsible for synthesis and packaging of lipids? The answer is endoplasmic reticulum. Well, the endoplasmic reticulum, it is network of membrane. The endoplasmic reticulum, specifically the smooth endoplasmic reticulum, it is responsible for synthesis and packaging of lipids and production of phospholipids and steroids. The Golgi apparatus, it is primarily involved in modifying and packaging proteins, while the lysozyme, they contains enzyme for intracellular digestion. So remember, this endoplasmic reticulum, it is just like a factory. So it is a factory where the proteins are made. The ribosomes, these ribosomes are the workers. And remember, mRNA is the instruction. So these products, they can be either used inside the factory or they can be sent out. Just like proteins can stay in the endoplasmic reticulum or be transported to the other part of cells or the body. So which of following it is responsible for synthesis and packaging of lipid? It's the endoplasmic reticulum. So remember, the Golgi apparatus, it is responsible for modifying and packaging the proteins and lipids, whereas the endoplasmic reticulum, it is involved in protein synthesis and lipid synthesis. The lysosomes, they are responsible for the intracellular digestion, and peroxisomes play a role in lipid metabolism. Coming to the next question. So which of following cells junction allow for direct communication and passage of small molecules between the adjacent cells? The answer would be gap junction. So what are gap junction? Gap junction, they are the communication tunnels between the cells. So these are the gap junctions. So they are just like the communication tunnels between the cells. So they are the channels that allow small molecules. So it will allow small molecules like ions or electric signals to pass from one cell to other. So gap junctions, they are like little tunnel that connects the cells, enable them to exchange information and coordinate their activities. So they are just like a cell phones. So gap junctions are like small cell phones that use cells to talk to each other. So they help cells share information, molecules, and even the electrical signals. So inside these gap junction, there are special proteins that connect one cell to another. So these proteins are known as connexin. So they are just like a phone number for these cell phones. These connexin, multiple connexin, they connect together to form connexin. So these are just like walkie-talkies that allow cell to directly talk to each other. So gap junctions helps with the transmission of electric signals between the nerve cells. So they also let the molecules and ions pass between the cells. The cell junction that allow for direct communication and passage of the small molecules between the adjacent cells, they are the gap junctions. Desmosomes, well, they are just like the sport welders. So they are specialized structures that will hold the cell together and provide strong mechanical connections. So the desmosomes use protein filaments to attach one cell to another. The tight junctions, they are like seal that prevents the leak between the cells. So they create a tight barriers between the cell, preventing the substance from leaking between them. Think of tight junctions as Ziploc bags, keeping things from spilling out. Hemidesmosomes, they are like anchors for the cell to their surroundings. So they are like anchors that keep the cells in place and maintain the tissue integrity. So the desmosomes, they act as sport welders. Camp junction allows for the communication tunnels between the cells. The tight junction, they act as a seal to prevent leak. And hemidesmosomes, they are like anchor that attach cell to their surroundings. So your answer would be gap junction here. Let us move to the next question. It was asked in one of your AIMS examination. RBC in the blood cells, they repel each other. Well, the repulsion of the red blood cells, it is due to the negative charge. So the negative charge on the carbohydrate moiety of the glycoprotein in the RBC membrane, particularly the glycophorin A, that is heavily modified by these sialic acid residues. So these negative charge group, they will create a electrostatic repulsion between the adjacent RBC. So that will prevent them from sticking together or aggregating. So that is important for maintaining the fluidity and flow of blood in the circulatory system. So you can just remember it by consider each RBC as a tiny donut. So on the surface of donut, there are something glycoproteins with negative charge, like the tiny magnets that push them away from each other. So keeping the donut from sticking together, so that your blood flows smoothly between the RBCs. All are repelling each other, staying separate and not clumping up. So that is important because it will help your blood move easily through the blood vessels. So that is important so that your body gets oxygen and nutrients it needs. So thanks to this tiny magnet charge on your RBC, that prevents them to clumping. So the answer would be C. So because of carbohydrate moiety of glycophorin A in RBC membrane that have a sialic acid residue, that is why the RBC in blood repel each other. So this question was asked in recent AIMS examination. Hope you got the explanation. A polysaccharides coating that cover the outer surface of many bacteria and allow the bacteria to adhere firmly to the structures is glycocalyx. So the option are pili, glycocalyx, fimbriae. The answer would be glycocalyx. So glycocalyx, it is just like a shield for bacteria. So it gives two functions. One is the protection and other is the adhesion. So protection, it will act as a protective coat around the bacteria. So it will so suppose Superman has a super suit. So this one, the glycocalyx, would act as a suit for the that shield the bacteria from the threats in the environment. Second, these glycocalyx also work as a glue, so that will allow the bacteria to stick to surface whenever, whether it's in your body or an object. So this stickiness, it is vital for bacteria to survive. So the answer would be glycocalyx. Coming to the next question, the coagulation time it is prolonged in. Well, the coagulation time it is prolonged in hemophilia. So hemophilia, we know it's a genetic order that would affect the blood clotting. So people with hemophilia would have deficiency of certain proteins that are known as clotting factors. And have a tendency to bleed excessively even from the minor injuries. So that leads to increased coagulation time, as the blood will take longer time to form clots. So the coagulation time, it's prolonged in a hemophilia. Coming to the next question, the pernicious anemia, it is caused by the deficiency of it is caused by the B12 deficiency. So pernicious anemia, it is caused by deficiency of the intrinsic factor. That is a protein. It is a protein that is formed in the stomach. So this intrinsic factor, it is responsible for absorption of which vitamin? Vitamin B12. So when there is lack of intrinsic factor, it can lead to inability to absorb vitamin B12. So that in turn would cause pernicious anemia. So vitamin B12, we know it's an essential nutrient your body needs for various functions, including making RBCs and maintaining a healthy nervous system. So when you eat food containing B12, it will go into stomach and your intestine. So intrinsic factor would play a crucial role here. So it acts as just a guide, as a tour guide, escorting vitamin B12 through the lining of your small intestine and your blood stream. Think of intrinsic factor as a guide that will lead you through a museum. So now if there is a problem with the intrinsic factor, the B12 cannot get through the lining of your intestine. So the tour guide is missing and not doing its job. So without enough B12, your body will not make enough RBC. So this shortage of red blood cells, that would lead to condition that is known as pernicious anemia. Anemia means that you have a fewer oxygen carrying red blood cells in your body. So that can lead to symptoms like fatigue and weakness. So hope answer is clear. Moving to the next question. The urea and creatinine excretion indicates early renal damage. Urea and creatinine excretion can indicate early renal damage. So increase in level of urea and creatinine in the urine may suggest that kidneys, they are not functioning properly, and there may be a kidney damage. So that can give an early indication of renal problems. The base of heart it is formed by, well, the base of heart it is formed by right atrium and left atrium. So the base of heart it is remember the top part of heart, and it is separated from the rest of thoracic cavity by structures like esophagus, aorta, etc. So think of heart like a house. The base of heart is like the roof of the house. It's the top part. Now the roof is mainly made up of left atrium, which is one of the heart's chamber. Coming to the next question, when a patient pulse is being taken, his arms should be well supported and above his shoulders. So extending and positioning at the same level or lower than that of heart. So this position is so that ensure a accurate measurement of the pulse. So placing the arm in different positions, they can lead to inaccurate reading. So the arm should be at the same level or lower than that of heart. Which of following contributes to the osmolality of the plasma? So among the options, the proteins they contribute to least to the osmolality of the plasma, while the glucose, sodium, urea, they helps to determine the plasma osmolality. So osmolality of plasma, it's measure how many concentrated or diluted your blood is. It depends upon things like what is dissolved in your blood, like salts, sugars, proteins and waste products. Among these, the proteins they contribute to least concentration, least of the osmolality of the plasma. The sodium it is the biggest contributor. Coming to the next question, the ECF volume of plasma. ECF volume it is determined by amount of sodium in the extracellular fluid. So ECF volume it is primarily determined by the amount of sodium in the extracellular fluid. So the extracellular fluid, we know that it's the fluid that exists outside of your cells in your body. These fluids would include the liquid part of your blood and the fluid that is surrounding your cells. So the question is, what determines the amount of ECF in your body? So plasma it is concentration of the plasma is not the primary factor that determines how much ECF you have. The amount of sodium you have in ECF, the more the sodium in your body, the more would be the ECF volume. So ECF volume is proportional to amount of sodium contained in the ECF and not to the concentration of these ions as per Guyton and Hall. Children and adults they have same concentration of sodium in plasma even though ECF volume are greatly different. The immunocytosis or reverse pinocytosis requires the calcium. So reverse pinocytosis, what do you mean by that? Let us understand this. Imagine a cell that has something inside a tiny bubble or vesicle and it wants to get out. So in a normal pinocytosis, the cell will bring things into this bubble, but in a reverse pinocytosis, they are pushing the things out. So the cell membrane, like the outer layer of the bubble, it fuses or connects with the vesicle covering. A small channel opens up between the inside of vesicle and outside of the cell. So the stuff inside the vesicle that wants to release passes out through this channel and goes out of the cell. So reverse pinocytosis, where the cell push material out of the vesicles, it involves the calcium. It involves the calcium ions. So inside cells there are vesicles containing substance that the cell want to release. To activate the release process, the cell need a signal. So here the calcium would act as a signal. So when the cell receives the signal, it allows the influx of calcium into the region where the vesicles are located. So this increase in the calcium is just like a green light for the release process. The potassium homeostasis in human is mainly maintained by the fact that most of the potassium ions are found intracellularly inside the cells. So that is a key factor in regulating the potassium level in the body. While the other options like active secretion of potassium in distal tubule or maintenance of sodium potassium ATPase activity also play in potassium regulation, the distribution is the fundamental aspect of the potassium homeostasis. So which cells stop the leakage from the vessels? So these are the platelets. So platelets as shown in this figure. So when the blood vessels they are injured, the subepithelium is exposed. So the platelets, the cells that prevents blood from sticking out of an injured blood vessels. So they will form a platelet plug and hence formation of fibrin leading to the clot formation. So moving to the question, the aldosterone action is on. Well, the aldosterone, it mainly acts on the distal convoluted tubule. So it causes the reabsorption of sodium ion in exchange of potassium and hydrogen ions. So let us discuss how it occurs. Suppose you have just finished a strenuous workout and you have lost a significant amount of sodium through sweat. So your body would need the right balance of sodium to keep your blood pressure and electrolyte in equilibrium. So your body would detect the decrease in the sodium due to the sweating. So that response that will trigger a series of events. So due to the decreased level of the blood sodium that occurs due to the sweating, there will be activation of renin-angiotensin aldosterone system. So the renin, it is released by kidney in a response to low sodium. So renin will convert angiotensinogen that is produced by liver into angiotensin 1. The angiotensin converting enzyme, that is this enzyme that is primarily found in the lungs, it will convert the angiotensin 1 into angiotensin 2. So the angiotensin 2, it is a potent vasoconstrictor and it would stimulate the aldosterone. It would stimulate the adrenal gland. So it will stimulate the adrenal cortex to release aldosterone. So now this aldosterone would primarily affect on distal convoluted tubule. So aldosterone would promote the reabsorption of sodium ions from the urine back into the bloodstream. So that would help to retain sodium that would be otherwise lost in the urine. So in exchange for the sodium resorption, the DCT excrete potassium and hydrogen ions through the urine. So that would maintain proper potassium and pH level in the body. So the reabsorption of sodium ions, it increases the water resorption. As more sodium is reabsorbed, water follows, which increases the blood volume and maintain the blood pressure. So in this way, the aldosterone, it helps to restore the balance of electrolyte and maintain the blood pressure where the sodium loss is high in case of the excessive sweating. So the aldosterone, it acts on the distal convoluted tubule. Hope your concept is clear now. Let us move to the next question. The macula densa, it is related with the distal convoluted tubule. The macula densa, it is related to this distal convoluted tubule. This one is the macula densa. So what is macula densa? Well, it is an area of closely packed specialized cells lining the wall of distal convoluted tubule at the point where the ascending limb of loop of Henle, it meets the DCT. So imagine you are playing sports on a hot day and you are sweating a lot. The sweat would contain salt. When you lose too much of salt, it can affect the body. So your kidney, they work to clean your blood, getting rid of the waste and keeping the right balance of things like the salt. So what macula densa would do here, inside your kidney there is a special spot that is known as this macula densa. What is the function of that? It is basically a salt sensor. So macula densa is a salt sensor. It will check how much salt is in liquid that the kidney is clearing. After you work out, you lose a lot of salt in your sweat. So the macula densa would sense these changes and it will send a signal, that is a alarm signal. So the alarm signal triggered by a substance called renin. So this alarm signal would trigger a substance that is known as renin, that is released by the kidney. So the renin starts a chain reaction creating something called angiotensin 2. So it squeezes your blood vessels, that raises your blood pressure. It tells your adrenal gland to make a hormone that is known as aldosterone. So this aldosterone would hold more salt and water and also helps to get rid of extra potassium. So macula densa, it is related to the distal convoluted tubule. That's your answer. Let us move to the next question. The passive stretch tension in the muscle, it is caused by the tightening. So passive stretch tension in muscle, it is primarily caused by the protein that is known as titin. Titin, it is a large protein that is found in the skeleton muscles. So imagine you are having a rubber band. When you stretch this rubber band, it resists and create tension, right? Now think of this rubber band as a simplified representation of muscle fiber and tightening. Think of rubber band as a single muscle fiber. So this one is a single muscle fiber. So when you stretch the rubber band, you are stimulating the act of stretching a muscle. That happens during activities like stretching your legs, arms, etc. So when you stretch the rubber band, it will create what? Tension. So this tension in the rubber band is similar to the passive stretch tension in the muscle. Now let us relate it to the titin. So titin, we know it's a protein that is in your muscle. It is a special type of rubber band within your muscle fibers. Each muscle fiber, it is made up of small units that is known as a sarcomere. So when you stretch your muscle, you are stretching the sarcomeres within the muscle fiber. So titin is like a special rubber band that plays a role in this process. When you stretch your muscle, titin resists and create a passive stretch tension. So it acts as a spring, storing the potential energy. So when you stop stretching the muscle, it relaxes the stored potential energy. It is released, helping the muscle return to its original shape. So this is essential part of the maintaining muscle structure and function during activities involving stretching and movements. Let us move to next question. In case of excessive ADH secretion, which part of the renal tubule it will show maximum hypertonic collection? So excessive excretion of ADH, that is also known as vasopressin. So that could lead to retention of water in the body that can affect the renal tubules. So ADH, we know it's a hormone that plays a role in regulating the body's water balance. It acts on kidney to control the amount of water that is reabsorbed from urine into the bloodstream. So excessive secretion of ADH primarily affect the collecting tubules. The collecting tubules, they are the final segment of the nephron in the kidney. So when the ADH it is overproduced, it makes the wall of collecting tubule more permeable to the water. The term hypertonic fluid collection means that solution with a higher concentration of solute. In this case, it means that fluid in the collecting tubules that becomes more concentrated when the water is reabsorbed. So by resorbing more water, the hypertonic fluid in the collecting tubule, it helps to increase the concentration of solute in the blood. So that can help to maintain the blood pressure and body fluids. So your answer is B. Cryoprecipitate, it is a rich source of, the cryoprecipitate, it is a rich source of factor 8. So cryoprecipitate, it is special part of blood that will contains the essential clotting factor. So imagine it as a life-saving ingredient for people who are troubled with the blood clotting. So think of cryoprecipitate as a magic potion for your blood. Some people have a condition like hemophilia, where the blood they cannot clot properly. So this means they can bleed a lot even from the small cuts. So inside cryoprecipitate there is a factor that is known as factor 8, right? That is like a superhero for the blood clotting. So person with hemophilia don't have enough of this superhero, that is factor 8, in their blood. Now imagine someone with hemophilia had a bad injury or needs surgery. The blood won't be able to stop the bleeding of its own. So doctors they can give them cryoprecipitate that is full of factor 8. So it is giving the missing superhero to their blood. So with the help of cryoprecipitate, now the person blood can now make more clots, so that would stop the bleeding. The packed red blood cells, they are prepared by taking whole blood and removing most of the liquid part, leaving mainly the RBCs. So the packed red blood cells, they are stored at temperature of -6 °C. So they are used when someone has low red blood cells level like anemia and need to improve their oxygen level. The fresh frozen plasma contains almost all the factors needed for blood clot, including labile factor 5 and 8. Fresh frozen plasma, it is stored at a low temperature at -18 °C. So cryoprecipitate, it is a rich source of factor 8. Clotting capacity of blood after storage, it diminishes. So when blood it is stored for at cold temperature, some changes occur over the time. Potassium, well, it is a key electrolyte that leaks out of the RBC. The pH of blood decreases, making it more acidic. Important compounds like ATP and 2,3-DPG diphosphoglycerate in RBC, they decreases. So that will affect the RBC ability to release oxygen to the blood tissues. WBC and platelets, they can degrade over time. Factor like factor 5, 8, they can deteriorate. So all these changes means that the stored blood, it is not as effective as clotting as the fresh blood. So the clotting capacity of the blood, it would diminish after the storage. So that is why the fresh blood or blood products, they are preferred in the critical situations. Coming to the next question, a patient came to you with systolic pressure of 190 and diastolic pressure of 100. His heart rate was 74 and pulse pressure was 90. Which of following would decrease to increase the pulse pressure? Which of following is a complication of diabetes? Well, the answer is all of above. The microvascular complications that involves the large blood vessels, conditions like coronary heart disease, peripheral artery disease, and cerebrovascular disease. So these conditions would increase the risk of heart attack, strokes, and circulation problems. Neuropathy refers to the nerve damage. Diabetes can lead to various forms of neuropathy that can affect both the sensory and the motor nerves. It can cause symptoms like tingling, numbness, weakness. Nephropathy means kidney damage. So the high blood sugar level can harm the small blood vessels in kidney that could lead to the kidney diseases. The early marker for renal damage in the known case of diabetes mellitus is microalbuminuria. So what do you mean by microalbuminuria? Microalbuminuria, it refers to presence of small amount of protein, that is albumin, in the urine. So that can be detected in the early stage of the kidney damage. So it is a widely used early clinical indicator for the diabetes nephropathy. So the early marker of the renal damage in case of diabetes mellitus is indeed the microalbumin urea. So presence of small amount of protein that is known as albumin in the urine. So in the diabetes, when the blood sugar level they are poorly controlled, small blood vessels in the kidney they can be damaged. So the microalbumin urea it is the first sign of this kidney damage. What does the term firing threshold in the reference to nerve impulse conduction mean? So firing threshold it is a level of depolarization. So it initiates rapid influx of sodium ions during the second part of the depolarization. So in context of the nerve impulse, it is when the transmembrane potential becomes sufficiently positive to trigger the opening of the sodium channels, allowing the rapid influx of the sodium ion. That is the second part of the depolarization in the action potential. So think of nerves as a tiny wires that carries the electrical signal in your body. So this signal is what helps you feel things, moves and do everything your body does. So now the nerve it has something that is known as the starting point for this electrical signal. We called it as a firing threshold. So it is like nerve's way of saying, okay, it's time to send the signal. So imagine your nerve is like a doorbell. The doorbell does not ring until you press the button hard enough. So if you touch it, it won't ring. But if you push it firmly, it will ring. So in the nerve, the firing threshold is like that hard enough. It is just like a hard enough push for the doorbell. So it is a level of excitement or energy the nerve need to start sending its signal. So when the nerve it will get excited enough and it reaches its firing threshold, it's just like the doorbell ringing. The nerve gates they open and where the magic begins. Just like the doorbell rings and you hear the sound the nerves send out of its signal. So this is where the sodium ion would come into play. They rush into the nerve, making the signal strong and allow it to travel down to the nerve. So when the sodium ion rushing, the signals start moving along the nerve. So the firing threshold is the nerve way of making sure that signal it is strong enough to start. And influx of sodium is what the signal ring and move along the nerve. Just like when you press a doorbell firmly, it starts to ring. So when a cell it is placed in hypertonic solution, so it shrinks due to the loss of water. It occurs due to the osmosis of water from cells into the solution. So when a cell it is placed in hypertonic solution, it will shrink due to the loss of water. Because the hypertonic solution has a higher concentration of solute particles compared to inside of cell. As a result, the water would move out of the cell through the process that is known as osmosis. So that will cause the water cells to lose water and shrink. So imagine a balloon inside a sugar jar. So suppose there is a sugar jar and you have a balloon. So think of cell as a tiny balloon floating in a jar of sugar water. So inside the cell there is a clear water like pure air inside the balloon. So now outside the cell there is a sugar water. So this sugar makes the water thicker and thicker. Now the sugar water in the jar is like a hypertonic solution. So water loves to balance things out. It's like to move from where there is lots of it where to less of it, so that it would make the water uh, to water balance. So when the cells in sugar water, they are hypertonic, the water from inside the cell, it starts moving out. So it's just like a air escaping from the balloon. As water leaves the cell, it starts to shrink, just like the balloon gets smaller when air comes out of it. So when the cell it is placed in hypertonic solution, the water will move out and cell would shrink. Moving to the next question, which of following is the classical example of multi-unit smooth muscle? The answer is pillow motor muscle. The uterus and gastrointestinal tract, they are example of single unit smooth muscle. The multi-unit smooth muscle is the pillow motor muscles. Pillow motor muscles, they are responsible for causing hair to stand on end, like goosebumps. They are example of multi-unit smooth muscle. Which of following is the linking or coupling agent between the excitation and contraction? The answer is calcium. So the coupling material between the excitation and contraction in the muscle, it is the calcium. Calcium ions, they are play an important role in regulating the muscle contraction. So when the calcium it combines with the troponin C, so let us discuss the muscle contraction process. So it all starts with signal from brain. When you decide to move a muscle, your brain would send an electric signal down from the nerve to the muscle. So there is a release of the neurotransmitter. So this neurotransmitter release acetylcholine. So that would send the signals to muscle to get ready to contract. It is just like flipping a switch to turn on the light. So inside the muscle cell, there are special storage cells, storage units that are known as sarcoplasmic reticulum. So they would release the calcium ions. So this is where the troponin C comes into play. The troponin C, it is a sensor. When the calcium they are relieved, the troponin C binds to them. So this binding is a crucial step in the muscle contraction. So troponin C, it will work with the other two proteins, tropomyosin and troponin 1. So they all sit on the muscle thin filament. So when troponin seeds bind to the calcium, it cause tropomyosin to move away from the binding site on the thin filament. So myosin heads, which are part of muscle thick filament, can now attach to these binding sites on the filaments. So it is like a handshake between thick and the thin filament that will form a cross bridge. So when myosin head pulls on the thin filament, they slide past each other. So that is actual muscle contraction. So as thick and thin filament, they will slide over each other, the muscle will get shorter. So it's the calcium that is the linking or coupling material between excitation and the contraction. A man is rushed to casualty, nearly dying after massive blood loss in an accident. There is not much time to match the blood group. So physician decided to order one of the following blood groups. The answer would be O negative. So O negative can donate blood to everyone. So O negative blood can be given to people with any blood type without causing any severe transfusion reaction. So it is the universal blood donor, as it lacks the AB antigen that can cause adverse reaction in the recipient. So the physician should decide to order O negative blood for the patient who has experienced massive blood loss. The normal pH of intracellular fluid is slightly acidic to extracellular fluid. So the normal pH of intracellular fluid is slightly acidic as compared to the extracellular fluid. The extracellular fluid is alkaline with pH of around is alkaline, while the intracellular fluids, they have a slightly lower pH. So B is your answer. Which cell organelle contain DNA? It's the mitochondria. The organelle that contain its own DNA is mitochondria. Mitochondria have their own genetic material that is important for your entrance exam point of view. Donan effect is due to, so Donan effect, also known as Gibbs-Donan effect. So that occurs due to the impermeability of the cell membrane to certain substance, particularly proteins and associate charges. So think of cell as a one room and extracellular space that is outside the cell as another room. So these rooms, they are separated by a barrier that allows only certain things to pass. So inside the cell there are larger proteins with the negative charges. So these proteins cannot easily pass through the cell. So they are like big heavy furniture that cannot be moved through the door. So in the extracellular space that is outside the cell, there are ions, both the positive ions and the negative ions. So these ions can move in and out of cell because the cell membrane, it allows them to pass. So the negative charge proteins inside the cell, they create a electrostatic attraction for the positive charge ion out of the cell. So these positive ions, they are attractive to the negative charge of the protein inside. So over time equilibrium is achieved. So the positive ions outside the cells, they are attractive to the negative proteins inside so they can only get so close to protein due to the impermeability of the cell membrane. So that will create an electric potential difference across the membrane. So Donan effect is crucial for maintaining the balance of ions inside and outside the cell. So it helps to ensure that the cell interior retain a specific electric charge and concentration of ions. So the Donan effect is a result of impermeability of cell membranes to protein, leading to the potential difference. So it is like a charge standoff between the ions outside of cell and proteins inside that will create an electric potential difference across the cell membrane. The permeability of cell membranes to ions, it is factor that plays an important role in Donan effect, but the key factor is the impermeability of the cell membrane to protein. So both of them, they are the answer. Thrombostatin is thrombostatin is a contractile protein that is involved in muscle contraction. So the mechanism of action of thrombostatin involves its role in platelet aggregation. That is a crucial step in blood clotting. So when a blood vessel is injured, platelets are activated and they come and form a platelet plug that will stop the bleeding. Thrombostatin, it is a part of process. So when the blood vessel is damaged, signals are sent to nearby platelets. So thrombostatin helps the platelets to change shape, allowing them to stick to the injured portion of the blood vessel. So this thrombostatin will promote the binding of platelets to each other. It's just like a glue that holds the platelets together and form a stable blood clot. So this aggregation of platelets is essential for forming a solid blood clot. So it is a contractile protein. The osmotic pressure it is determined by the total number of particles, both solute and the solvent in solution. So more the particles there are in solution, the higher will be the osmotic pressure. So it is proportional to total number of solute and solvent present in the solution. Substance that helps in linking the cytoskeleton of cell to plasma membrane is the ankyrin. So ankyrin, it is a protein. So remember, ankyrin is a protein. It plays a role in linking the cytoskeleton of cell to the plasma membrane. Specifically, it helps to attach spectrin, that is another cytoskeleton protein, to the plasma membrane. So this linkage, it is essential for maintaining the structural integrity and the proper function. Ankyrin, it is a protein that is found in red blood cells. It helps to attach spectrin to the cell membrane. It acts like a glue that holds the spectrin in place and make sure that spectrin skeleton is properly connected to cell membrane. So ankyrin, it is important for maintaining the red blood cell shape and stability. The maximum surface area, it is present in the capillaries. So capillaries have the maximum surface area. So the extensive surface area of capillary is essential for effective exchange of gases, nutrients, waste products between the blood and the surrounding tissue. So this vast network of the tiny blood vessels would provide a large area for this exchange to occur. Which of following is most characteristic feature of smooth muscle? So one of the main characteristic feature of smooth muscle is absence of the transverse striation. Option A, central plate nucleus. Well, this feature is associated with the cardiac muscle and not the smooth muscle. In cardiac muscle, the nuclei are centrally located, which is the feature that distinguishes it from skeletal muscle. Autonomous innervation. Autonomous innervation is a feature of both smooth and cardiac muscle. The autonomic nervous system controls these muscles, where the controls these muscles involuntarily, whereas the skeletal muscle, they are primarily under the voluntary control. Absence of transverse striations. This is the most important characteristic feature of the smooth muscle. Unlike the skeleton muscle, smooth muscle lack the transverse striation. Which of following is the most characteristic feature of smooth muscle? The answer is again the absence of the transverse striation. connective tissue sheath surrounding the muscle as whole unit. It's the epimysium. So this one is the epimysium. It is the connective tissue that surrounds the entire muscle. Endomysium, it surrounds the individual muscle fiber, and perimysium, it surrounds the bundle of the muscle fiber. The single most important factor in control of automatic contractility of heart is the sympathetic stimulation. So think of heart as a pump that pushes blood throughout your body. The heart need to adjust its speed and how hard it pushes depending on what you are doing. So suppose when you are just sitting or sleeping, your heart beats at certain rate and with a certain amount of force. But when you are running or doing strenuous exercise, your body would need more oxygen. So your heart here will be pump faster and stronger to deliver oxygen quickly. So this adjustment in your heart is like a car engine, adjusting to go faster when you press the gas pedal. So in your body, this adjustment is controlled by something that is known as sympathetic stimulation. So it is like someone stepping on the gas pedal for your heart. So when you are running or stressed, your body sends a signal to step on the gas, and your heart feels faster, stronger to meet your body demand. So answer would be sympathetic stimulation. Megaloblast, they are the precursors of normoblast. Megaloblast, they are the early stage cells in the development of the red blood cells. As they mature, they progress through the different stages, including early, intermediate, and late normoblast, before finally becoming the reticulocytes and then the mature erythrocytes. The fluid content of blood, the fluid content of blood is the plasma. Plasma, it is the liquid component of blood that make up the largest portion of the blood composition. Plasma contain water, electrolyte, proteins, hormones, waste products, and various other substances that are essential for the proper functioning of the body. The most important plasma protein is the albumin. Albumin, it is a protein that is found in highest concentration in the blood plasma. It play a role in maintaining the oncotic pressure of the blood. So albumin, it plays a role in regulating the force that is known as oncotic pressure. So think of oncotic pressure like a sponge that will attract and hold on water. So this force would keep the fluid inside the blood vessel and prevent it from leaking into the surrounding tissue. So albumin help to maintain and create this pressure by drawing water into the blood vessel. Other function of albumin is, we know edema, it is accumulation of excessive fluid in the blood in the body tissue. So albumin helps to prevent the edema by keeping the fluid in the blood vessels. While the albumin levels are normal, it acts as a sponge, pulling the fluid into the bloodstream. Albumin also serves as a transport molecule, carrying the various substance throughout the body. That includes the hormones, minerals, calcium, and magnesium. So it helps to maintain the overall volume of blood in the circulatory system. The most important plasma buffer, it's the bicarbonate. The absolute refractive period of neuron is the phase of depolarization. So think of a nerve like a messenger that will carry important messages in your body. To send a message, the nerves would need a special language that is known as action potential. So action potential is basically the nerve's language. It is like a nerve way of talking. It is an electric signal that runs along the nerve. So when the nerve it wants to send a message, it starts an action potential. So before sending a message, the nerve is in a resting state. It's like a quiet phone line before you make a call. At this point, the nerves have certain electrical balance. So when the nerve wants to, when there is depolarization, it's time to send a message. The nerve balance changes. So that is known as depolarization. So it is just like a picking up phone and starting to talk. So the nerves here would get excited. And third is the action potential. It is when the nerve is actually sending the message. It is like a conversation on the phone. So the action potential travels down the nerve carrying the message. The step 4 is the absolute refractory period. So now here when the busy part comes in. So right after the action potential there is a special time that is known as absolutely refractive period. So during this message the nerve is like someone who just finished conversation and can't talk another call. So why break? So the absolute refractory period, it is a safe measure. It is actually a safe measure. So it ensures that action potential has a time to move in one direction and not get more signals, so that the action potential has time to move in one direction and it does not get cope up with the more signals. So it's just like a traffic light that keeps the road clear for one car at a time. So absolutely refractory period, it is a phase of depolarization. So after the absolute refractive period, the nerve it can go back to its resting state and be ready for the next message. So absolutely refractive period is a phase during which neuron is entirely unresponsive to any additive stimulus. So it coincides with the rapid depolarization phase of the action potential, which is when the neuron electrical charge is rapidly changing. So the absolutely refractive period, it is relatively short, that lasts for around 0.5 to 1 millisecond. The relative refractive period, it occurs just after the absolute refractive period. So during this phase, the neuron can respond to another stimulus, but it requires a more stronger stimulus. So the refractive period, it is primarily due to the fact that some of the potassium channels remain open, making it easier for the potassium to leave the neurons. So the absolute refractive period, it lasts for around 3 to 5 millisecond. So absolutely refractive period is phase of depolarization. That was your answer. Which of following is the outer part of the nerve? It's the epineurium, already discussed. The muscle spindle would detect changes in the muscle length. So primary action of the muscle spindle is to detect the changes in the muscle length. So when your muscle is at rest in certain position, it is not sending any special signals. Now imagine when you decide to stretch your muscle, like you extend your arm or leg. So as you stretch the muscle fibers would also start to stretch. The muscle spindle is sensitive to the stretching. When it detects that the muscle fibers are getting longer, it will send signals to your brain. So these signals travels to your brain. Your brain receives the signals and process them. So it can decide whether to make adjustment in the muscles activity to accommodate the stretching and maintain the muscle control. So muscle spindle detects the changes in the muscle length. So remember, change in the muscle length that is detected by muscle spindle, whereas the change in muscle tension it is detected by the Golgi tendon organ. The substance that is present in both serum and plasma, it's the factor 5. Plasma, we know it's a liquid part of the blood that contains various proteins and substance including clotting factors like factor 5. So factor 5 is present in both plasma and serum. Teichoic acid, it is present in the cell wall of primarily present in the cell wall of gram-positive bacteria. And in gram-negative, the cell wall lacks typically the teichoic acid. Pernicious anemia occurs due to the deficiency of due to failure of secretion of intrinsic factor. Already discussed, this question was already discussed. The answer is B. Increase in the cytosolic calcium during smooth muscle contraction, it is due to the IP3 DAG, that is inositol triphosphate and diacylglycerol. So in smooth muscle contraction, the increase in the cytosolic calcium plays a crucial role. So this increase in the cytosolic calcium because of it occurs due to the two mechanisms. The first is the influx of the calcium ion, they enter the smooth muscle cell through the voltage or ligand gated plasma membrane channels. Second is the release of calcium from the sarcoplasmic reticulum. Inositol phosphate, it is a signaling molecule that triggers the release of calcium from the sarcoplasmic reticulum, that is a storage site for calcium within the cell. So this release is facilitated by IP3 receptors that are present on the sarcoplasmic reticulum. So they play a role in signaling the release of calcium from the internal stores of smooth that causes during the smooth muscle contraction. The cytosol structure, it is maintained principally by the microtubules. The cytoskeleton, it is a network of protein filament within the cytosol. The cytoskeleton, it is essential for maintaining the cell shape, providing structures, providing the support and facilitating various cellular processes. So that consists of three components: microtubules, microfilaments and intermediate filaments. The microtubules, they are made up of tubulin protein that provides the framework for the cell. It helps to maintain the shape of cell. So microtubules, they are crucial part of cytoskeleton and they are responsible for maintaining the structural integrity and shape of the cell. Increased aldosterone leads to all except. Increased aldosterone, they can cause hypernatremia. So there is increased level of sodium in blood. We know that aldosterone, it promotes the reabsorption of sodium in the kidneys that can lead to higher level of sodium in the blood. It can cause hypokalemia, that means the lower level of potassium in the blood, It causes aldosterone. It will cause excretion of the potassium in the urine, reducing the concentration in the bloodstream. It causes hypertension. Increased aldosterone can lead to high BP. That can cause this is because the aldosterone causes the kidney to retain sodium. That can lead to fluid retention and increase blood volume. Metabolic acidosis, it is characterized by excess acid in the body. Increased aldosterone, it is not associated with metabolic acidosis. It is associated with metabolic alkalosis. So that's your answer. The white muscle fibers, they are fast twitch. So white muscle fibers, they are type of fast twitch. We have two types of fibers, type 1, type 2. Type 1 are slow fibers twitch, slow twitch. Type 2 are fast twitch. So white muscles are fast twitch. Slow twitch, type 1 are red because of myoglobin and mitochondria, whereas the fast twitch, they are white due to absence of myoglobin and mitochondria. So fast twitch fibers, they are known for their ability to contract quickly, and they generate a lot of force. So answer is A. The blood vessel in normally do not clot because this question was discussed earlier also. The inner lining of blood vessel, it is called vascular endothelium. So this endothelial layer, it is smooth and has special coating that is known as glycocalyx, that is made up of charged polysaccharide rich proteins. So glycocalyx, it is highly charged and plays a role in preventing the blood clotting. Valerian degeneration, it is found in the peripheral nerves. So Valerian degeneration refers to the degeneration of nerve segment distal to the site of trauma. So suppose this one is the point of injury. So there will be degeneration of the nerve segment distal, that is farther away from the point of injury. So this procedure, it occurs in the peripheral nerves, the nerves that are located outside of the brain and spinal cord, those in your limbs, etc. In Valerian degeneration, the part of nerve located beyond the injury starts to break down. If the extracellular sodium concentration is reduced, then so action potential, we know they are signal that allows nerves to communicate with each other. So these action potential, they have characteristic amplitude, that is essentially the size of the electric signal. So the amplitude of action potential, it will depend upon the balance of the positive charge ions like the sodium inside and outside the nerve. So when an action potential is generated, the sodium ions rush into the cells, causing the cell membrane potential to become more positive. So this is what the action potential happen. So that makes the action potential to happen. So if you reduce the concentration of extracellular sodium, that means there are lower sodium levels, sodium ions outside the cell. So it means it becomes more challenging for the sodium ions to enter the cell during the action potential. So with the fewer sodium ions entering the cell, the amplitude of action potential is reduced. So answer would be C. The Golgi tendon organs, we discussed already that they will sense the muscle tension. and muscle length is by the muscle spindles. In an isotonic contraction of muscle, the muscle moves through a load through a distance. So isotonic means the muscle contracts uh, sh- and shortens. So isotonic contraction is when the muscle contract and it's as it contracts, it actually moves a load or a object. Think of lifting a weight like a dumbbell in as your bicep muscle contract, it shortens and moves the weight through a distance. That is isotonic contraction. In isometric contraction, when the muscle generate force without change in the length. So the muscle is contracting but it is not moving a load or there is no change in the length. So example is pushing against a immovable wall. That is example of isometric contraction. So fatigue in the skeletal muscle, it occurs due to the depletion of the ATP that is necessary for the muscle contraction. So when the ATP it becomes low, the muscle can cannot no longer generate energy for sustained contraction that leads to the muscle fatigue. So the accumulation of lactic acid is a factor in the muscle fatigue, but it is more commonly associated with the short duration, high intensity exercises rather than the low intensity exercise. So we know that the lactic acid it is build up of lactic acid buildup. It is a byproduct of anaerobic metabolism and it contributes to the muscle fatigue during intense activities. Moving to the next question, the red muscles it consists of. Well, the red muscles they consist of higher concentration of myoglobin. So these red muscles, they have high content of myoglobin and cytochrome with many mitochondria, rich blood supply, uh, and they are smaller in diameter. So the red muscle it consists of high concentration of myoglobin with oxidative fibers that gives it a red color. We know myoglobin it is a protein that stores the oxygen in the muscle cells and red muscles they are also known as slow twitch muscle. Red muscle fiber they are also known as slow twitch muscle slow twitch fibers. So they contains more myoglobin to support prolonged contractions. So that is why they appear red in color due to the myoglobin content and increased blood supply. So they have a increased blood supply. That is why they appear red in color. They contain a higher number of mitochondria that are responsible for the aerobic energy production. Red muscles they have a few, whereas the red muscles they have a rich blood supply. Whereas the fast muscles, they are also known, the white muscles, they are also known as a fast twitch muscle fibers. So they are involved in activities like sprinting, weight lifting, etc. Coming to the next question, in smooth muscle fiber, the calcium binds to. Well, in the smooth muscle cells, the contractile process involves binding of the calcium to the calmoduli. So the answer is B. So the first step in the smooth muscle contraction is increase level of the calcium. So there is increase in intracellular calcium level that can be triggered by various signal including transmitters, hormones or change in the membrane potential. So when the intracellular calcium concentration it rises, the calcium ions will bind to protein that is known as calmodulin. So this calmodulin it act as a calcium binding protein. So when calcium binds to it, it undergo a conformational change. So binding of calcium to calmodulin, it forms a complex that is known as calcium calmodulin complex. So this complex it is important for the smooth muscle. This process it is important for the smooth muscle contraction. So the calcium calmodulin complex it activates enzyme that is known as myosin light chain kinase. So this myosin light chain kinase it is responsible for phosphorylation. That is, it add a phosphate group to the myosin cross bridges within the smooth muscles. So once it get activated, uh, it allows myosin to bind to the actin filament and initiate the sliding filament mechanism leading to the muscle contraction. Coming to the next question, during BP measurement with sphygmanometer. So when measuring BP with sphygmanometer, it is important to ensure that the cuff and the device they are properly positioned to obtain accurate reading. So placing the cuff and sphygmanometer at the level of heart helps to ensure that measurement it is not affected by the gravitational differences between the heart and arm. So that can lead to inaccurate reading. So both cuff and sphygmanometer should be at the level of heart. Now the P wave in ECG recording it is caused due to the the P wave. So this P wave is caused due to the atrial depolarization. So P wave it would represents what? Atrial depolarization. That is the electrical activation of atria. So it corresponds to the initiation of the electric impulse at the SA node. So that spreads through the atria leading to atrial contraction. The other is QRS complex. The QRS complex uh, indicates the ventricular depolarization. So that is the electrical activation of the ventricles, that is the lower chamber of the heart. It includes Q wave, R wave, and S wave. So that leads to ventricular contraction. The other is the T wave that represents the ventricular repolarization. That is the recovery of the ventricular muscle cells following the depolarization. So P wave would represents the atrial depolarization. The SI unit of measurement of BP. Well, the SI unit of measurement of BP, it's kilopascal. Well, the pressure in SI unit, it is measured in pascals. BP in SI unit is measured in kilopascals. And 1 mm of mercury is equal to 0.133 kilopascal. That is the conversion. Hematocrit is defined as Hematocrit, it is defined as the percentage of blood volume that is occupied by the erythrocytes, that are the red blood cells. So it is important measurement of concentration of red blood cells in the blood, and it is typically expressed as percentage. And in normal male, the value of hematocrit, it is around 45%. So hematocrit, it is a blood test that will measure the volume percentage of red blood cells in the whole blood. So it provides information about the proportion of the red blood cells in the blood. So a low hematocrit would suggest anemia that could be through nutritional deficiency, chronic diseases, bone marrow diseases, whereas the high hematocrit uh, may indicate polycythemia, that is condition that is caused by increased number of the RBCs, that can be due to the chronic lung diseases or certain smoking or certain bone marrow disorders. What is a serum? Serum is plasma minus fibrinogen. So serum is the plasma minus fibrinogen and other clotting proteins. So when blood, it is allowed to clot and clot retracts, it releases serum, which is initially the plasma without protein involved in blood clotting, that is without fibrinogen and other clotting factor. The venous return to heart during quiet standing, it is facilitated by all of following factors, except the answer would be D. The gravitational, we know the gravitational increase in arterial pressure does not enhance or improve the venous return during quiet standing. In fact, the gravitational, it causes an increase in arterial pressure. It can be considered a potential hindrance to the venous return. So gravitational increase in arterial pressure, it would cause hindrance to the venous return. So when the person, it is upright position, standing position, The gravity can increase the pressure within both the arteries and veins in the lower extremities. So this increased arterial pressure does not directly facilitate the venous return. So it can make it more challenging for the blood to flow from vein in the legs to back to the heart. As the blood have to work against gravity. So answer is D. Vitamin K dependent coagulation factors include. So remember this mnemonic. Uh, 2 plus 7 is 9. 2 plus 7 is 9 and not 10. So the mnemonic, the vitamin D dependent clotting factor are 2, 7, 9 and 10. Hope it is clear. All of following cells, they are connective tissue region except the Clara cells. So the Clara cells, not of uh, they are not of connective tissue region. So these Clara cells, they are restricted mainly to the terminal and respiratory bronchioles. They are cuboidal, non-ciliated cells. Coming to the next question, the membrane protein clatharin. Well, the membrane protein clatharin, it is involved in receptor mediated endocytosis. We know clatharin, it is a protein that play a key role in receptor mediated endocytosis. So this process involves the selective uptake of specific molecules from the extracellular fluid by the cells. So the clatharin coated pits forms on the cell membrane, and these pits are coated with the clatharin protein. So the receptor mediated endocytosis, it is essential mechanism for cells to internalize specific substance like nutrients and signaling proteins by binding to receptors on the surface cell and then engulfing them in a vesicle coated with clatharin. The in a primary in the primary active transport, the covalent bonding with pi causes conformationally modulation of carrier protein. So primary active transport, it is a type of active transport in which the energy it is used directly to transport the molecules across the biological membrane, typical against their concentration gradient. So the energy source for primary active transport, it is usually ATP. So which energy is used? ATP. So a common example is the sodium potassium ATPase. So sodium potassium pump is its example. So sodium potassium pump, it is a protein that is found in the cell membrane of uh, most animal cells. So it actively transport 3 sodium out of the cells and 2 potassium inside uh, cell for each ATP molecule used. So this pump, it is essential for maintaining the proper balance of sodium and potassium inside and outside the cell. So in the sodium potassium pump, the pump binds to intracellular sodium. The ATP, it is hydrolyzed to ADP, and the energy that is used, released during this process, it is used to change the shape of that of the pump, that is conformational modulation. So this change in shape that will allow the pump to release sodium outside the cell. So the pump then binds to extracellular potassium ions. So another conformational change is induced that allows the pump to release sodium, potassium inside the cell. So the covalent bonding with pi, it will cause conformational modulation of the carrier protein. In the secondary active transport, the movement of sodium is from higher concentration to low concentration. So the secondary active transport, it is also known as it is also known as co-transport. So there are two types of secondary active transport. One is symport. In symport, both molecules they move in same direction, whereas in antiport, the molecules they move in opposite direction. The example is sodium glucose co-transport. So that is responsible for the transport of glucose against its concentration gradient in the renal tubules of the kidney. So the example of secondary uh, glucose transport is the sodium glucose co-transport that is found in the renal tubules of kidney. So it is responsible for the reabsorption of glucose from urine back into the bloodstream. So in the extracellular fluid, there is high concentration of sodium due to the primary active transport by the sodium potassium pump. So SGLT will bind both the sodium and glucose on the luminal side of the renal tubules. So the binding of the sodium induce a conformational change, allowing both the sodium and glucose to be transported into the cell. So this movement of sodium from high concentration to low concentration gradient, it is driven by sodium potassium pump. So once inside the cell, the glucose it can be transported across the basolateral membrane in the bloodstream via facilitated diffusion through the glucose transport. So the answer is A. The nerve conduction, it is the nerve conduction is an active self-propagating process that requires energy for the propagation of nerve impulses at constant amplitude and velocity. So the active conduction it is facilitated by the movements of ion, specifically the sodium and potassium across the cell membrane. Donan effect is due to, answer is D. We have already discussed this question back. The isovolumic relaxation phase of cardiac cycle it ends with the opening of the AV valve. So isovolume relaxation, also known as isovolumetric ventricular relaxation. So it is a period in which both the aortic and pulmonary semilunar valves are closed. So during the isovolumetric uh, relaxation, the ventricles they are relaxing, and the pressure within them it is decreasing. So the AV valves that separates the atria from the ventricles they remain closed due to prevent in order to prevent the backflow of blood from ventricles to the atria. The semilunar valves that guards the exit from the ventricles into aorta, they are also closed. So the isovolumic relaxation period it ends with the opening of the AV valves. So when the pressure in the ventricle it drops below the pressure in atria, the AV valves they get open, allowing blood to flow from atria to the ventricles. So this marks the transition from the isovolumic relaxation to ventricular filling phase, during which the ventricles they are filled with blood from the atria. The QRS complex would indicate, already discussed, it would indicate ventricular depolarization. When a normal person changes position from standing to lie down, the following changes is seen. So when the normal person position changes from standing to lie down, the venous return to the uh, heart, it increases almost immediately. So in the standing position, the blood can pool in the veins of lower extremity due to the effect of gravity. So when the person lie down, this pooled is returned to the central circulation, increasing the venous return of the heart. So this venous return helps to maintain the cardiac output and blood pressure when changing position. During diastole, the arterial pressure it is maintained by during diastole, the phase of cardiac cycle when the heart is relaxed and it is filling with blood, the arterial pressure it is primarily maintained by elastic coil of the aorta. So during systole, the heart pumps blood into aorta and large vein, stretching their walls to accommodate the incoming blood. So when the systole ends and diastole begins, the heart relaxes. So during this relaxation, the elastic walls of aorta tends to returns to their original size. So this is known as elastic recoil. So as the wall of these elastic vessels will recoil, they will exert pressure on the blood inside, helping to maintain the continuous forward flow of blood and arterial pressure during the diastole. The best method to assess the adequacy of replacement of fluid in case of shock is increase in the urine output. So increase in urine output indicates that uh, kidneys they are receiving sufficient blood flow and are capable of filtering and excreting waste products and excess fluid. So it is a quantitative and relative reliable indicator of organ perfusion. So in case of shock, maintaining or increasing the urine output, it is positive sign that circulatory system it is responding to the fluid replacement. The urine output it is a practical and reliable method to monitor the response to the fluid replacement in case of shock, as it will represents the kidney overall function organ perfusion. A synthetic antidiuretic hormone that controls bleeding in dental patient with the type 1 bone Willebrand disease, it's the desmopressin. So it is a synthetic antidiuretic hormone that is control used to control the bleeding in patients with von Willebrand disease. So it helps to improve the platelet function and enhance the blood clotting. The cardiac muscle they have a the cardiac muscle they have a long refractory period. So that means that after the cardiac muscle fibers contracts, it takes long time before it can contract again. So during the refractory period, the muscle fiber cannot respond to another stimuli, no matter how strong it is. So a long refractory period of cardiac muscle, it is essential for the proper functioning of heart and its ability to pump blood effectively. SA node is known as the pacemaker of heart because of fact it generates impulses at higher rate. SA node is also known as pacemaker of heart because it possesses the ability to generate electrical impulses at highest rate. So remember the SA node, it generates electric impulses at the fastest rate among all the components of the heart electrical conducting system. So SA node, it is the natural pacemaker of the heart because it is capable of generating impulses spontaneously, and it does so at highest rate, setting the pace for the heart rhythm. The increased venous return to heart, it will cause uh, increase in end diastolic volume. So end diastolic volume, it is the amount of blood in the ventricles of heart at the end of filling phase, just before they contracted during systole. So imagine a person who has been resting for a while. In this state, the body oxygen demand is relatively low, and veins they are returning blood to heart at a steady state. So this person and diastolic volume is considered baseline. Now, what happens during exercise? Increase venous return when person starts to exercise. The muscles are working harder and require more oxygen. So this increased muscle activity that results to increase in return of the deoxygenated blood to the right atrium. So the increased venous return means that there is more blood filling the right atrium during systole. As a result, the right ventricles receive a large volume of blood during its filling phase. So this increased blood volume in the ventricles at the end of diastole, it is reflected as end diastolic volume. So the heart responds to increased diastolic volume by increasing the force of contraction during the systole. So that allows the ventricles to pump out larger volume of blood that is known as stroke volume. So increased venous return as during exercise, it leads to increase in the end diastolic volume. That in turn contributes to increased stroke volume and the cardiac output. The arch of aorta, it begins at the level of sternal angle, that is also known as angle of Lewis. The right ventricles they receive blood from the right ventricle they receive blood from the superior vena cava. So superior vena cava, It's a large vein that carries deoxygenated blood from the upper half of body back to the heart. So blood from the superior vena cava flows into right atrium. From the right atrium, the blood then passes from the tricuspid valve into the right ventricle. So the right ventricle, it is responsible for pumping this deoxygenated blood into the pulmonary artery that carries it to lungs for oxygenation. The membrane protein clathrin it is involved in, answer is D. Already discussed. The majority of the blood sodium it is present in the extracellular fluid. So sodium is the most abundant cation that is present in the extracellular fluid. QRS complex indicates the ventricle depolarization. Already discussed. The isovolumic relaxation phase of cardiac cycle it ends with the opening of the AV valve. Already discussed. Which of following is not a sarcolemmal protein? The answer is D. Sarcolemmal proteins, they are proteins that are found in sarcolemma, that is the cell membrane of the muscle cells. So these proteins they play a role in muscle structure, functioning and communication. Lectin it is a large multi-domain proteoglycan that is found in extracellular matrix. It is not a part of sarcolemma or the sarcolemmal protein. In human, the least used physiological response to low environment temperature is the pilorectin. So pilorectin refers to phenomenon where the small muscles at the base of hair follicles they contract, causing hair to stand on the end. So this is also known as goosebumps. It is the physiological response to cold or emotional stimuli. So that occurs due to the sympathetic nerve activation. So it is commonly it it does not provide it is not very useful in the humans. So the least useful response to low environment temperature is the piloerection. Other options like shivering, it is a mechanism by which the body generate heat by rapidly contracting and relaxing the skeletal muscle that helps to increase the body temperature. Vasoconstriction that refers to the narrowing of blood vessels in response to cold, which helps to reduce the heat loss by decreasing blood flow to the skin surface. And thyroxine, it plays a role in regulating the body's metabolic rate and can be increased in response to cold to generate more heat. The heme it is converted into bilirubin mainly in the spleen. The breakdown of heme to bilirubin it occurs in the macrophages of the reticuloendothelial system that occurs mainly in the spleen as well as in liver. So answer is spleen. The blood supply of SA node is right coronal artery. The cell junction that allows exchange of cytoplasmic molecules between two cells, they are the gap junctions. Already discussed, this question was already discussed before too. The distribution of blood flow is mainly regulated by arterioles. Arterioles, they are the major site of resistance to blood flow in the circulatory system. So by changing their diameter, arterioles can significantly influence the resistance to the blood flow in the different vascular beds. So that determines how much blood is delivered to various tissues. So option B is correct. Which of following can be determined by calculating the clearance of para-aminohippuric acid? The answer is effective renal plasma flow. So para-aminohippuric acid, it is commonly used method to estimate the effective renal plasma flow. So the rate at which para-aminohippuric acid is excreted in urine, it reflects the rate of plasma flow to the kidneys. So this is an important ERPF. It is important parameter for evaluating the kidneys function, and it is used in the determination of renal blood flow and glomerular filtration rate. Which of following statement is true about the troponin C? Troponin C, the is one of the main component of the troponin complex. So it binds calcium ion, and in presence of calcium it initiates the muscle contraction by permitting the interaction of actin and myosin. So when the calcium, it will binds to the troponin C, it will cause a conformational change in the tropomyosin complex, allowing the myosin to bind to actin and starts the contractile process. So it permits the interaction of actin and myosin.